Hi, everybody. This is the should be the last review recording for the first exam, and we left it after I had talked a little bit about valence electrons and things like that. This is getting into the bonds. Remember all the differences. Nonpolar typically has carbon to carbon, carbon to hydrogen bonds. Okay, ionic bonds, opposites attract, positive and negative charge. They form salts. Again, salts are very stable physically, but very reactive chemically because they dissolve rapidly in water. Sodium and chloride, sodium with a positive, chloride with a negative. The interesting thing about hydrogen bonds is it has to do with that sort of tug of war I demonstrated on the board on several occasions. You have oxygen and hydrogen, nitrogen and hydrogen, phosphorus and hydrogen, and sulfur and hydrogen. That's why that periodic table's sticking up there. It's basically demonstrating that because of the sort of asymmetric, irregular placement of the electrons, okay, in the corner, it basically allows the oxygen, the nitrogen, the phosphorus, the sulfur to take over and pull those in such a way that effectively we create polarity. Pulling of the electrons makes more negative on the oxygen, nitrogen, phosphorus, and sulfur side, and on the hydrogen side, more positive. Hence, polar covalent bonds. And what that basically creates is what we call hydrogen bonding. That amount of polarity is not enough to basically, how should I say it, to basically cause them to separate most of the time, with the limited exception of water. But what it permits, interestingly, is those basically the polarity to come in. So hydrogen bonds can form. Now those hydrogens have weak attraction to other things. And you remember looking at DNA, looking at water, those little dots, those are hydrogen bonds. A very, very weak magnetic attraction. And the more of those NHs, OHs, pHs, and SHs that you have basically makes a molecule, even a larger molecule, like glucose, okay, with a lot of OHs, being effectively what we call bigger part, polar covalent bonds. So hydrogen bonds are sort of the foundation for this concept of polar covalent bonds, which is different than this carbon to carbon and carbon to hydrogen thing. And I hope that's clear. And if it's not, please shoot me an email. And to amplify that point a little bit, while I see what's on the email situation, okay, text situation, is effectively what pH is, the partial dissociation of water, that teeny tiny amount. But that's the amount of water that effectively, despite the hydrogen bonds, is able to just a little bit destabilize and create H pluses and OH minuses. They're equal in water because it's neutral. Anything you add to the water that releases more protons, hydrogen pluses, is an acid. Anything that makes less of them available proportionately is called a base or an alkaline, or alkali would be the appropriate term. Once again, monomers and polymers. Spend a lot of time on this. ATP is a small molecule. It's the monomer. Okay. Adenine, these are nucleotides. Adenine, guanine, cytosine, thymine, uracil, small molecules. They're the building blocks of RNA and DNA. So even though it looks like it's got a lot of stuff, comparatively speaking, it's a small molecule. It's not mammoth like DNA or RNA, ATP. Glucose is not mammoth like starches and glycogen. Those have just chains upon chains of glucoses together. So we have small molecules as the monomer, and then the polymers, amino acids, small molecule. But when you chain, hundreds of those together, now you can get a protein, or maybe less than 100, you have a polypeptide. That's the whole idea, is understanding the difference between a small molecule and a macro or a large molecule. That's really the importance. So the building blocks of small molecules, simple sugars, monosaccharides for starch, carbohydrate, and glycogen, amino acids for polypeptides and proteins, those respectively are the polymers, and nucleotides like ATP for DNA and RNA. That's just the way it works. And again, they're formed by dehydration synthesis. We pull out of water and voila, there they go. So effectively, when we want to break them down, we add water. That's what we do in our bloodstream or in our gut. More probably appropriately, we have a better place to look at it. Again, amino acids are characterized by that central carbon. It's got, remember, four different areas to bond. And it's bound with four different things: an amino on one side, an acid on the other, a hydrogen on the other, and that R group that has, again, one of 20 things. And we went over that. The idea between essential amino acids and the 20 total amino acids that we utilize biologically, 8 or 9, depending on the author, are essential amino acids. And peptide bond forms between the amino and the acids on adjacent molecules. So that's another thing to be familiar with. And then all those structures of proteins. Remember, hydrogen bonds make up the secondary structure. Okay. And they can either be spirals or pleated sheets. The globules or fibers are tertiary structures. That has to do a lot with the R groups. Remember, acids and bases, positives and negatives, water solubles and fat solubles. And again, when you do the denaturing thing, when you expose it to temperature or heat, temperature or pH variabilities or other things that can cause the protein to denature, to lose its shape, or to change its shape so it no longer has the same function. That's denaturing it, losing its very nature. You end up with one thing left: the primary structure, basically the batting order of the amino acid sequence. That's the key thing. And again, know the breakdown of these things. Know the monomers: glucose, fructose, and galactose. The dimers, just add a glucose. Glucose and glucose is maltose. Glucose and fructose is sucrose or table sugar. Glucose and galactose, okay, is lactose. Famously, that galactose tells you it's lactose. And so what their structure is, those are 6-carbon sugars. Ribose and deoxyribose are 5-carbon sugars. The oddball is that glycerol. That's a 3-carbon alcohol. That's what we see in triglycerides, where it gets its name in fats. Again, the big difference between glycogen, starch, and cellulose, they're all made out of glucose. The problem is the way we can digest the branching molecule, which is glycogen and starch, but cellulose, it's cross-linked. And the idea that we don't have the enzymes like cow would have, who can graze in the field, we can't. Okay? It doesn't mean it's not utilizable, because that's what dietary fiber comes from. All those cell walls and other building blocks inside cells that are structural elements we can't digest, but we can utilize different kinds of fiber, both soluble and insoluble. Insoluble fibers are bulk agents for your digestive tract. Soluble ones pick up molecules like cholesterol type fatty molecules in a process called adsorption, ADSORPTION, not absorption. That's sort of a combination. So it's a bit different. Remember the structure of a nucleotide: a nitrogenous base, A's and T's, G's and T's, ribose or deoxyribose, a 5-carbon sugar known as a pentose, and a phosphate. That's the minimum requirements for nucleotide. ATP is the oddball because it specifically has adenine, ribose, and has those 3 phosphates, and that's sort of the magic with those similarly charged particles that, in order to hold that molecule together, you have to impart extra energy, hence the idea of a high energy bond. That's very, very significant. It all has to do with the similarities in charge. Enzymes, by definition, lower activation energy. Again, reminder for the composition of the most important fat: triglycerides, a glycerol, and 3 long-chain fatty acids, 8 to 20 carbons, or sort of the construction materials about that. When we make a phospholipid and eventually make the membrane, we take away one of those enzymatically. In other words, we have a reaction that occurs in our body. We take one of those fats, we remove a long-chain fatty acid, and we replace it with a phosphate. And then other things can attach to the phosphate. And so when it has all the hydrogens, saturated fat, saturated, meaning everywhere there can be a hydrogen, there is one. If we have sites of unsaturation, we have a double bond there instead. And again, when we look at it with the natural thing that those phospholipids do, again, I've said it again and again recently in the last couple of lectures: if we put Phospholipids in water, what do they naturally do? They form that membrane, that phospholipid bilayer. The hydrophilic heads on the outside, the hydrophobic tails on the inside, okay, on the interior of the phospholipid, and they effectively create the membrane. And you have water-soluble molecules, the heads, protruding, if you will, outside and inside. One side of the membrane, the other side of the membrane, that's the compatibility. I think that's everything I have to share with you about this. Again, email with any questions. We'll see everybody on tomorrow, which is Wednesday. If you're listening in today, which is Tuesday the 23rd. Thanks again for your attention.